Hello, everyone, and welcome to our webinar entitled Tips for a Successful Study in Medical Aesthetics, presented by Innovaderm Research in collaboration with our partner, QuantifiCare. Innovaderm is a full-service CRO headquartered in North America and dedicated to both medical and aesthetic dermatology clinical research. QuantifiCare is a full-service imaging partner of Innovaderm, headquartered in France with extensive experience in both aesthetic and medical dermatology studies. Next slide, please. My name is Jason Shuris. I'm the Chief Business Officer here at Innovaderm. I have been in this role since August of this year, and I've been with the company almost 3.5 years in total. Next slide, please. Our participants today are our esteemed panel who you'll be hearing from talking about medical aesthetic trials. From left to right, we'll begin with Dr. Jasmina Jankicevic, the Chief Medical Officer here at Innovaderm. Dr. Jankicevic is a Doctor of Medicine, Master of Medical Sciences, and is a board-certified dermatologist and venereologist. She's a renowned keynote speaker, trained journalist, and author of over 150 articles in peer-reviewed trade journals. She's a key opinion leader in dermatology drug and device development, as well as medical affairs. In addition, she's led global clinical development and medical strategy in dermatology and medical aesthetics for both CROs, pharmaceutical companies, biotech companies, and cosmetic companies. Her work has led to the implementation of over 300 clinical trial protocols for more than 20 different medical and cosmetic dermatology indications. In addition, we have Mr. Nicolas Dapis, the Chief Business Officer at QuantifiCare. Nicolas graduated from both French and Canadian business schools and has 17 years of experience in medical imaging in hospitals and in clinical trial settings. Last but not least, Nalanie Johnson is a project manager here at Innovaderm. Nalanie has been with us for almost 2 years and has been in the industry for over 10 years with extensive experience in site engagement and management experience in aesthetic dermatology research. She holds a Bachelor of Science in Human Biology and a postgraduate certificate in clinical research. And without further ado, I will introduce you to our first speaker, Dr. Jasmina Jankicevic.
Thank you, Jason, for that really kind introduction, and warm welcome to all of you, dear colleagues around the world. It is wonderful and great that you've decided to join this session that will be focused really on how can we best design and implement clinical studies in medical aesthetic space. I will focus on actually looking into strategic as well as scientific and medical considerations that are of paramount importance to all of us who are on the quest to develop in medical aesthetic space. I will not spend too much time introducing myself because Jason did such a great job. Probably the most interesting news of a sort for you is that I've joined Innovaderm several months ago, and my role is the role of Chief Medical Officer. With that, let's move to the next slide. Really, everything starts in building the strategy, starts with alignment between clinical development thought and knowledge of the market. And when I say knowledge of the market, I first of all mean understanding the market in the context where are the unmet patient needs. So if we look at the medical aesthetics market, the growth is absolutely amazing. And medical aesthetics seem to be bulletproof even compared to many other quite successful markets that have experienced the challenges during the COVID pandemic. In terms of worth, in 2022, we're talking about close to $14 billion that will really jump to almost $24 billion in next 5 years. We also know that when we look at the regions, the #1 region is North America, and it will continue to be, followed by Europe. And in terms of fastest growing markets, APAC is #1, followed by Latin America. Why is global medical aesthetics market basically growing at such impressive speed? Some of the answers are captured in the this blue rectangular that you have on your right-hand side, and I will actually touch upon each of these in more detail in following slides. But what I would like to emphasize on this slide is our number one challenge in medical aesthetic space. That challenge is related to clinical risks and complications. They're primarily stemming from inadequate, unskilled application of drugs and device in medical aesthetic space. And the other segment of complications is derived from the fact that some of these drugs and devices are not coming from controlled, well-controlled and overseen manufacturers or through the right channels. Basically, they're coming from the black market, and consequently, the significant number of complications related to use of drug and device in medical aesthetic space has been caused by that particular fact. On the next slide, you will see that if we can move, there we go. On the next slide, you will see further elements of the answer to the big question, why such a significant growth and why does it matter to us as clinical developers? It is important to us because it's an opportunity for every company, strategic opportunity that will actually be crucially incorporated into how we design and implement clinical trials. It is very clear that in especially the last 2 decades, we've experienced continual increase in awareness and adoption and demand for medical aesthetic services. When you have increased demand, what happens? Well, our colleagues, healthcare practitioners, notice it, and they decide to jump in and offer some of these medical aesthetics services. As a consequence, right, having these services being more accessible, you will have even higher adoption of some of these services. That increase in adoption is also demonstrated through adoption in some new patient segments. So what we've seen, especially in the last 10 years, is increase in application of some of these medical aesthetics procedures among geriatric population in last 5 years, especially in millennials and male population. So this is like a perfect positive feedback type of cycle that we are experiencing. Together with that, what is happening is development of advanced technologies that are implemented in the core process of developing these new solutions, as well as producing them or mass producing them. And that has led to really accessibility to user-friendlier solutions. And what do we mean by user-friendly? And this is what we need to keep at the top of our mind as we're designing our new solutions and creating protocols for them. So we're talking about easiness of use. We're talking about downtime being as short as possible. We're talking about increased safety, and all of that packed in the context of very competitive effectiveness. This has actually spilled over not only from in-office procedures, but it's also been quite represented among home use devices. We've seen significant increase in development of these and a huge jump in use of those in recent years, again, due to pandemic. One of the most interesting market that most of developers are actually focused on are actually non-invasive and minimally invasive medical aesthetics procedures, because they're epitome of the less pain, more gain type of approach. When we say less pain, there are those user-friendly solutions, and obviously they're known to have less downtime, easier to apply, whether they're being in the office or something that is continued as a treatment at home. The final segment that makes our medical aesthetics opportunity even bigger is the fact that medical aesthetic space has accessed the precision medicine concept big time, which means that these days healthcare practitioners are developing elaborate treatment protocols that are going to be highly specific to certain patients, patient and that patient needs. As a consequence, they're combining many procedures. So this is an opportunity for all of us developers to be part of that new treatment paradigm. On the next slide, please, we're going to take a look at some of the key trends. And I've used the data from the Aesthetic Society from last year, so quite fresh. And what we see here is that top 5 minimally invasive procedures and top 5 surgical procedures are the one that we're all very familiar with. Why is that the case? Because the companies who are behind these particular procedures are continually investing in this particular space, and they're developing newer and more competitive solutions. So overall, this list is not a big surprise to us. If we would compare the data from, let's say, 5 years ago or even 10 years ago, some of the same elements or the same procedures are being represented, with the neurotoxins and dermal fillers on the minimally invasive side. And these are the ones that we're going to put in focus in terms of protocol considerations on the next few slides. But in the surgical space, we do see a little shift between liposuction, who used to be in the second place, and breast augmentation, who actually ruled this space for many years in the past. Now, the very new and exciting aspect of the trends is regenerative aesthetic medicine. Regenerative aesthetic medicine is still in its infancy, we should say, but the list of different opportunities, and I put on the slide 5 top areas or approaches that are being investigated, is truly exciting. Now, in the beginning, there were a lot of concerns about how is this space going to be regulated, but I'm sure that you're aware that in the meantime, FDA has issued 2 guidances. One is related to actually standards in regenerative medicine that is applicable to aesthetic space. And the other one is actually specifically focusing on regulatory considerations for cell and tissue therapies, meaning also cell-based and tissue-based procedures. In EMA realm of influence and responsibility, they've also issued new guidance for advanced therapy, where actually regenerative aesthetic medicine solutions will be considered. Now that we've understood where the market sits, this is the strategic part. So when we're thinking about how are we going to design our study and how to make the study optimally impactful, but at the same time feasible, we're actually going to move to the nitty-gritty detail of protocol considerations. I mentioned to you that because the most excitement is still in dermal fillers and neurotoxin space among minimally invasive and non-invasive procedures, that's what we're going to focus on today. So in dermal fillers world, the world was shaken, especially by the increased number of adverse events reports that led in early 2021 to a panel meeting discussing general issues with dermal fillers organized by FDA. I would encourage you to actually look for it. The executive summary as well, the slides from that panel meeting are publicly available. But what came out is the significant additional confirmation of what should be our protocol considerations. And FDA really has one of the highest regulatory standards. So if you're meeting those, you're probably going to be completely equipped to be considered by regulatory agencies in other regions. From a study objectives perspective, safety is always going to be in the core and in the focus. As you know, one of the main considerations and issues that we had with dermal fillers are intravascular injections that can lead to blindness, but can also lead to stroke. Hence, huge focus from the regulator's perspective on safety. In terms of how you will approach effectiveness, you will have 3 different approaches. One is it's going to be sort of a preliminary effectiveness, or shall we say feasibility type of a study for completely new fillers. Your focus on effectiveness is going to be much higher if you are, and easier in a way, if you are developing filler that is already approved, but you're developing it in the new indication. And then finally, the third type of objective could be a non-inferiority study, where your job is going to be to compare with the existing approved dermal filler. In order for your study to really give you the answers, the questions that you're asking, you need to define your patient populations and the circumstances of the study very well, which is all encompassed in inclusion-exclusion criteria. Number one is going to be, who are your patients? So the baseline severity of the defect, whether those are finalized in wrinkles or is it volume loss, needs to be well defined. What you will usually use is the same validated clinician reported outcome assessment tool that will help you determine the severity and decide whether the patient belongs in the study or not. The second very important aspect is to have the adequate representation of intended population. As you know, regulators have become increasingly sensitive to it, and FDA is clearly outlining that we do need to have at least 20% of the patients who are darker Fitzpatrick skin types, those being from 4 to 6, and actually at least 10% of the patients who are Fitzpatrick 5 or 6. The second segment on this list are actually exclusion criteria. Obviously, we don't want to include in the study patients who have recent treatment or concurrent treatment in the region that we're focusing on. However, on the other side, if we're, for example, treating and investigating the effect of dermal fillers somewhere on the face, if the patients had an aesthetic treatment on the abdomen, that patient is really not to be disqualified, right, and excluded. The other important thing is to eliminate patients with all underlying conditions that may, first of all, increase the safety concerns, but on the other side also confound your study. And then finally, as I mentioned at the beginning, one of the biggest concerns is the potential negative effect and adverse events related to ophthalmic system. So any type of ophthalmic conditions that can actually blur the ability to assess the safety of the dermal filler really need to be excluded from the study. The interesting part about dermal fillers is that you have more options for how to control your study. So you can choose to have a non-treatment control, you can have a sham treatment, or you can actually have an active comparator. So it's an active placebo, an active control. Safety assessments, big focus on them in general. So the injection site reactions as well as the treatment responses need to be very diligently tracked in what is called a safety patient diary for at least 2 and up to 4 weeks after the injection, right after the application of the dermal filler. In addition to that, adverse events need to be closely reported and recorded throughout the study. What we are looking at is incidence, we're looking at safety, meaning the severity, we're looking at duration, we're looking at relatedness, as well as the resolution. Has the resolution happened? When did it happen? Or if not, what have been a follow-up? In addition to that, you have to focus on indication-specific assessments. For example, if we're treating lip area, lip movement, ability to speak. If we're treating hands, do we have a normal mobility of the hands? If we're treating cheeks, can we smile and move these areas normally? So all of that needs to be part of the safety assessments. And then finally, the most sensitive part is making sure that there are no visual impairments. And there is a whole segment actually in the executive summary of the panel meeting that I mentioned to you that is outlining everything that needs to be done before the patient is enrolled, after the treatment is applied, actually where there are particular assessments that need to be performed 30 minutes after the application, and then obviously follow up and continued check-in. Related to safety assessments, the language needs to be very clear also in informed consent form about the potential adverse events related to vision. And that is a very significant expectations from both regulators as well as ethics committees. In terms of efficacy assessments, your primary endpoint will be assessed by use of the validated clinician-reported outcome. You will most often see these days that regulators are recommending to have a validated PRO as a co-primary. And obviously how the data will be reported, it's going to be reported as a responder rate. And what regulators are interested in is being able to show a clinically significant effect of the treatment. The last thing that you should know is that you need to have a repeat treatment as a part of your protocol. So the safety and effectiveness of the second treatment have to be assessed, usually anywhere between 6 and 12 months, but it's going to depend obviously how you're projecting the duration of the effect and peak of the effect of your particular dermal filler. And that is very important because this is the real life situation, right? In the real life, our patients will come back for the second treatment, especially if they're pleased with the treatment, they see the effect and they have no safety issues with it. And the last segment to it is to really make sure that you understand when the retreatment is to be timed. In the next slide, we're focusing on botulinum toxins, and protocol considerations in some areas are actually overlapping. So what I decided to do here for you is kind of just outline the main ones that are applicable to your considerations for phase 2 program, and then some that are for phase 3 program. In the phase 2 program, first of all, it is very important to identify safe and effective dose. So this is going to be your dose ranging study. The goal in the phase 2 program is to very carefully and deeply uncover any potential dose-related side effects, because that will dictate the choice of the dose in the phase 3 program. And finally, this is the time to investigate: is your delivery system working? Are there any technique issues that you should be aware of? And you will inherit those learning and incorporate them into your protocol language in the phase 3 program. In the phase 3 program, you will be expected to deliver 2 double-blind randomized placebo-controlled trials. You will again be expected to have a good representation of the patient population that the botulinum toxins is intended for, which means inclusionary representation, having all Fitzpatrick skin types. The one question that I often get when I consult for my colleagues that are outside of the United States and that have decided to actually develop outside of the United States, please be aware that your data may have actually be viewed differently by FDA if you're interested in being approved in the FDA. And what FDA is highly likely to request is that you have US executed a clinical trial or a segment of the clinical trial. In terms of effectiveness, you will be expected to have a co-primary. Co-primary is going to consist from a clinician-reported and patient-reported outcome that are supposed to be validated. Most of the companies actually do internal validation. In most of these cases, this is applicable for fillers and also botulinum toxin. Photonumeric scales are used as a clinician reported outcome. And in some cases, the language can be adjusted and those can be used also as a patient reported outcome. In terms of safety, obviously everything related to adverse events as well as the laboratory data needs to be supplied. But in addition to that, for fillers, we don't have as much of that issue, but for botulinum toxin, it's about potential local or distant spread of the botulinum toxin that really needs to be looked into during the treatment and post-treatment. Same as for fillers, you will be expected to show the repeat dose within your trial, and to show when that repeat dose should happen and what are the safety and efficacy implications of the second dose. The regulators prefer when you will be expected to deliver a long-term trial, but regulators prefer that that trial be controlled versus open label extension. And then finally, for botulinum toxin development, you will be expected to show what is happening in terms of immunogenicity and how is that affecting the safety and the effectiveness of your particular botulinum toxin. What I did here is I've covered protocol considerations without mentioning one big ticket item, which is photography. Photography is of a paramount importance in development of medical aesthetics solutions, from perspective of being used in the trial as a tool to assess the patient progress and change in patient status, but also as a tool to use for a central assessment, and finally as a tool that you will use strategically throughout the life cycle of your medical aesthetic solution. And that is why we have our colleague from QuantifiCare today, Nicolas, who is going to talk to you about importance and the way to properly use photography in aesthetic trials.
Thank you very much, Jasmina, for the very nice introduction. Okay, so today's presentation is about photography in aesthetic trials. So as Jasmina explained in our great presentation, the efficacy of aesthetics treatment is assessed by using validated scales, which represent different aging severities. So these assessments are performed locally by blinded investigators. And sometimes that can be an issue, especially in multi-center clinical trials, because you can have some inter or some intra-rater variability. One way to deal with this issue is by taking photography at each site and have them securely transferred in central location and have a blinded reviewer reviewing the whole photography database in a randomized fashion and have the scales integrated. And this for sure is a better approach because you have one qualified central reviewer, reducing for sure the inter-operator variability. And as an alternative, you can still use a panel of experts with an adjudication process. The intra-rater variability can also be assessed by using by adding duplicates, for example, into the image database. And at last, this guarantees the fully blinded process because the image are randomized and the reviewer is blinded not only on the treatment, but also on the treatment time points. So when assessing the pictures, the expert doesn't know if he or she is assessing baseline or follow-up pictures. So for this to work, you have to make sure that you're taking highly standardized photograph. And this is really a challenge in aesthetic clinical trials, especially at the site level, because you need to make sure that the sites have a photography setup, a professional photography setup with a uniform background, with some professional lighting. And this is clearly a burden for the clinical sites because you need to have dedicated space, a room allocated sometimes for photography procedure. You need to make sure to use professional camera systems as well. That can be complex. And you need to make sure that you are using also optimized settings to have a good image quality. And that's also is a challenge. You need to make sure that patients are carefully prepared for the photography portion, meaning that you need to make sure to remove makeup, jewelry, make sure that patients are de-identified before taking any picture. And again, this is an extra burden for the clinical site, meaning that it's taking extra time for them. And at last, that's the image acquisition part, because you need to make sure that pictures are very reproducible, not only from a visit to another, but also from all the sites, across all the sites participating into the study. So if you take all these challenges together, there are not only limits to this central evaluation linked to the fact that we're required to have highly standardized photograph, but this is also linked to the central evaluation itself. Because clearly when performing central evaluation based on photograph, there's a lack of objectivity here. You need to have, there's a qualitative assessment which is performed with some human bias. In addition to this, these assessments are performed by using these validated scales, which are a non-continuous scoring system, meaning that even for a qualified central expert, sometimes very challenging to put a grade on a subject between 1 and 2. You cannot put 1.5, unfortunately, and you have to choose. So for all these reasons, 3D photography is definitely the best alternative, because one of the main advantages is that the 3D systems can generate some 3D standardized views to see pictures from an accurate angle. And all these pictures are then very reproducible, and then they can be assessed centrally. Another advantage of 3D photography is that it can be used for precise and accurate quantitative assessments, meaning that in dermal filler studies, for example, when you want to measure the global volume changes, you can accurately measure these volumizing effects of the dermal fillers. So there's a wide range of different camera systems available on the market. There are systems which can capture the face. There are systems that can capture the body. There are even systems to capture the microstructure of the skin. The idea when using 3D photography in aesthetics trials, to make sure you have a system which is easy to use, but also which is compact. Easy to use by using a system which is pre-calibrated with which capture perfect quality pictures by, for example, using dual beam pointers which overlap at the optimal distance. So you have very good looking pictures, very good quality pictures, which are also reproducible because these two dual beam pointers overlap at the same distance from a visit to another. The system has to be portable, compact, to make sure that it's not taking too much room within the clinical center. Some examples of pictures that can be captured and then some analysis that can be performed is, for example, by capturing the microstructure of the skin. There's a lot of different quantitative measurements that can be performed to evaluate, for example, the roughness of the skin, to measure the volume, the depth of one single wrinkle. So that can be used, for example, for cosmetics treatments, for neurotoxin like botulinum toxin studies, for example. Another possibility, another system is a system enabling to capture the full face. And the idea here again is in dermal filler studies, like volumizing products, with the ability to have the before and after pictures perfectly matched, to have a volume map of the difference as presented here. And we can compute and measure very precisely the volume difference between two visits. By reconstructing the whole face, there's also other type of analysis that can be performed, like, for example, the lifting effect of a treatment, with the ability to measure the lifting effects and to measure the length of vectors, which are then created and measured by using the system. And last but not least, by using the system for a body contouring treatment, the idea is to measure global volume changes either when you create volume on a procedure like a buttock augmentation, and you want to measure the positive volume that has been created, or also to measure the volume reduction like on the abdomen, for example, which is the fact here. So I would like to conclude this presentation by in our experience, the regulatory agencies are now starting to accept these 3D measurements as primary endpoint in their studies. And this is mainly the case for studies occurring, for example, either in Europe or in China. But for now, this is still very challenging in the US because the FDA is still considering validated scales only and local assessment as part of the primary endpoint. So hopefully this will change in the future. Thank you very much for your time. Now I would like to hand over to Nalanie for our presentation about the operational aspects of this aesthetics trial. Thank you very much.
Thank you so much, Nicolas. Hello, everyone. We can go to the next slide, please. As mentioned earlier, my name is Nalanie Johnson, and I've had the privilege of working in aesthetics over the years, and I would like to share some tips and traits that I've learned along the way so that we can have some consideration as we think about developing studies in this space. Let's go to the next one. The first thing I want us to think about is the investigative site. So choosing the right site for this kind of study is quite important. It's important mainly because we have to think about the kind of participants that's coming into this space and that's wanting to do trials in this space. So unlike medical dermatology, these participants aren't necessarily sick. They're not looking for medical treatment, but instead they're looking for enhancement. So the sites have to understand the patient population. They also have to be able to deal with influx when it comes to recruitment. So oftentimes in this space, recruitment goes quite quickly because as we put out ads and as we look for people to do this kind of trial, there is lots of participants that is going to be interested. So the sites have to have a setup so they can handle the influx. So that's one thing. So the resource is important. Understanding the population is another consideration to think about when you are looking for the right sites is sites who are able to understand that these patients are oftentimes coming during their lunch break or before work or after work. So being able to open later or start earlier is quite important to accommodate these patient needs. And this goes right in hand in talking about patient engagement. So as we select the right sites, these sites are going to know how to recruit these patients, not only recruit them, but retain them. So we don't want to lose our data. So we want to be able to retain our patients for the long haul. Some of these trials can take sometimes 2 years, especially if we're doing retreatment. So we want to make sure that we retain our patients in the study so that we can collect as much data as possible. And that goes to say that we want to ensure that our communication is open, not so much with the site and the participant, that's important, but also with the sponsor, the CRO, and the site. We want to ensure that we're keeping the sites engaged. We're letting them know when enrollment is going to be ending. We want to make sure that we don't have situations that the patient comes in for a visit and the database has been closed because we reached our target. So we want to keep our communication open with the sites, not only during the enrollment phase of the study, but throughout the entire study. Whether we're using newsletters or email blasts, but we keep the sites engaged so that we can keep our patients in the study. Another consideration that we want to take into account is our timelines. So as we begin to establish our timelines, we want to ensure that the sites are aware, that they're on board, that they know exactly what it is that they need to do so that they can keep our patients engaged and keep things going. So in a nutshell, having the right sites and having a strategy that we select the right sites who understand patients in this space is quite important. Let's go to the next slide, please. This slide is quite important because it touches on exactly what our friends from QuantifiCare was talking about. Two of our main outputs from this is going to be our photos as well as our PROs. So when it comes to photos, we want to ensure that the technology that we're using is quite easy for the sites to use. If they need to train their patients on anything, it's quite easy, so that the burden on the site is reduced and they're not spending too much time troubleshooting. That also goes hand in hand with the technology that we choose to use for our PROs. One of the things that we want to ensure is that patients in this space, because they may not have done research studies before, then there need to be some hand-holding or training from the site to the patient in how to answer these PROs or on what to do. So oftentimes, because it is very visual in nature, we want to ensure that the research staff have no influence on the patients when it comes to them doing their PRO. So those things are done first, away from any investigator or site staff, so that there's no chatter that can distort what they say on their PROs, because those are two critical endpoints that we want to ensure it's a lot of quality that's being maintained there. We can go to the next slide. So when we think about our study and we think about how we're going to be able to operationalize our study, some things can impact that. And one of those things can be the science behind that, or the novelty behind the molecule. So as we know, oftentimes investigators are very intrigued by different kinds of science, right? So we want to ensure that we have their buy-in, because they're interested in novelty of a product. So oftentimes that helps them to recruit more for us, because they're interested, they're engaged with us. Another one could be a competition, right? So sometimes there are other companies that may be doing the exact same study. So because of that, we want to ensure that we take a look at the database and see who's doing what, so that there's no competition for either the sites that we're trying to get to do our study. So that way we're able to have a smooth study. Another one that Dr. Jankicevic talked about earlier was the inclusion criteria. So ensuring that we have a flexible protocol, ensuring that our protocol is not too restrictive in terms of the procedures that we're doing. So although patient safety is quite important, we got to be careful not to put in too much study procedures. So things, for example, like doing a biopsy or doing excessive blood work may not be something that's appropriate for this kind of study because of the nature of the study. Another thing is a time of year. Oftentimes I find in my experience, patients are really interested and wanting to join a study based on when the effects may be seen. So if the effects are going to be seen close to the holiday period, they tend to be really interested in wanting to participate in the study, because they know they're going to have some nice results to show off, So it's really important to plan the study so that perhaps we can get some effect around the holiday periods. I touched on procedures earlier, so I won't talk on that one again. But I also touched on reducing the site's burden. So having a well-written protocol, a well-designed protocol, is really important for us to be able to run our study, and also having the right equipment that doesn't need a lot of troubleshooting. It's really important for our sites to be able to run our study. So now I'm going to pass it back to Jason, who's going to go give some key learnings.
Thanks so much, Nalanie, and thank you, Nicolas and Jasmina as well. I hope our guests today learned something from at least one, if not all three of our participants. Just to kind of summarize what we learned today. So from a medical perspective, I think it's very clear that medical aesthetics will continue to be an area of expansion in dermatology. There's a lot more technology coming to market and coming to the clinic. And Innovaderm is at the forefront of the research of that technology for sure. As far as study design and protocols are concerned, Obviously, the only feasible study protocols that align perfectly with regulatory requirements will give us a chance to generate timely and relevant data and successfully support the submissions for your both your medical and aesthetic drug and device trials. From a photography perspective, we learned the advantages of a central reading setup in minimizing the intra and intervariability of the assessments that are being performed in these studies. Of course, the photography also needs to be very highly standardized, which presents additional challenges, of course, but working with a group like QuantifiCare, who has done this hundreds, if not thousands of times in the past, really goes a long way towards achieving that standardization. And then, of course, 3D photography is a very good alternative to produce these highly standardized views and quantitative measurements. And it was very positive to hear that regulators are starting to accept and adopt this as a primary endpoint in a very standard way to get new approvals into the market. From an operational standpoint, selecting the right sites, as it is in medical studies, in aesthetic studies is no different. Selecting the right sites is extremely important to a successful study execution and high quality data. And then, of course, a very well-written and flexible protocol will allow for a rapid patient recruitment, which will, of course, lend to faster data in the hands of the sponsors and faster timelines to approval with regulators. So I hope everyone was able to learn something today. We have a couple of poll questions that we'll be asking you. I'll talk very briefly about Innovaderm in general, and then we will wrap up with a few questions if we have time. So the first question here is, did you know that Innovaderm is a full service CRO that also manages studies in Europe? The second question is, which of these criteria do you consider the most important when selecting a photography vendor for your trial? Relevant experience, price, reputation, or some other option? I'll finish off the presentation talking very, very briefly about Innovaderm, and then we'll get to some Q&A with our participants. So Innovaderm, as I mentioned previously, is a full-service dermatology-focused CRO. We are, in fact, operating in both North America and Europe. For those of you that answered yes to the first poll question, you are right. We've done extensive trials in both North America and Europe, over 150 trials in total. Innovaderm also operates a research site in Montreal, Canada, and that site has conducted more than 500 trials in both dermatology and healthy subjects. Our network of investigators globally consists of over 3,600 around the world, and our current operations team is up over 380, almost 400 folks right now within Innovaderm, full-time employees in both North America and Europe. And as I just mentioned, we do run trials globally, North America, Europe, and in some cases, Asia Pacific when the need arises through CRO partners. So now we have a few questions popping up. Let me see what we have here. So the first question is for Jasmina. Jasmina, the question is, do intra-patient designs have any merit in development of products in medical aesthetics?
Yes, they have actually quite a wide area of potential use, especially in early stages. So in your proof of concept segment, that's definitely one area where we see a lot of obviously publications and a lot of work done in that intra-patient design. The reason for that is you're eliminating any of the inter-patient differences related to the quality of the skin, to level of, let's say, photo damage of the skin, etc., when you are actually using the same patient. And you will also notice that during the life cycle management of your product, so if the product is approved and you're considering a new indication, what your investigators, key opinion leaders in the space will tell you is they get these ideas what new indications could potentially your medical aesthetic solutions be considered for, and they will tell you we could do, and usually they will tell you, a split face or split body study just to get the first sense and the first signal whether your filler or your neurotoxin or your device, your laser, your basically solution could be working in another area. So I would not dismiss them. But having said that, in your pivotal trials, especially for drugs in medical aesthetics, this is not going to be something that will be looked upon with absolute delight by regulators. However, the situation may be different when you are developing certain types of devices.
Great, thanks so much, Jasmina. The next question is for Nalanie. Nalanie, the question is, why is it particularly important to focus on patient engagement in aesthetic trials?
Yes, so it's important because understanding the patient population and understanding what the patient's needs are allows us to be able to recruit and retain those patients.
Great, thanks so much, Nalanie. The next question, let's get one for Nicolas. Nicolas, the question is, how long does it take to capture photos during a patient visit?
Well, it depends on if this is just standard photography. It can take, as you can see, there's a lot of different steps. So it can take up to 10-15 minutes through regular photography. Well, by using 3D photography, the idea is to capture and to spend no more than 3 up to 5 minutes max when using 3D photography with standardized snapshots to capture the patient's face, for example.
Great, thank you, Nicolas. And while I have you, I'll ask you another one. How do you ensure anonymization of these photographs?
That is a very good question. It is either performed at the site level before uploading pictures on our portal. Sites have the ability to veil images. There are also ways to automatize that process, and this is this is performed by the team, by QuantifiCare team, and this is the images are reviewed to make sure that there's no personal information appearing on the photographs.
Wonderful. Jasmina, another one for you. You mentioned that unmet needs should be one of the key considerations in clinical development strategy. In your opinion, what are the top unmet needs in aesthetics?
Wow, that's a big question. I'll try to actually keep it objective, but luckily in many aspects, what is probably objectively needed is what I would also like to see more of in terms of the development. But as a clinical developer, you can approach this from multiple segments. So the one thing is to talk about particular indication, right? Because that is an unmet need in itself. And if we're looking from that perspective, I would probably say scars are the #1 unmet need. I don't think we were able to fully tackle scars. And when I say scars, I mean acne scars, but I also mean atrophic, hypertrophic scars and keloids. So I think that's a big area of unmet need. But also, if we look at any region of the face, and especially if we look at the body, there's still many indications that could be successfully or more successfully tackled. But what I want to turn your attention to is, because we have a significant segment of development, let's say in fillers and neurotoxin space, this is where you need to think in the terms of unmet need in the context of what is already available. So how can you be better for the patient? What can you comparatively bring to the patient doesn't have right now? Is it a longer duration? And I would definitely agree that this is still unmet need. Having, for example, neurotoxins with a longer duration, or having neurotoxins where there is no concern about immunogenicity, or that concern is minimized. So after repeated uses, you have continued safety and efficacy profile. Or is it, when it comes to filler, again, a sustainable effect? So the distance between the two treatments is prolonged. Or is it that we have less concerns about the safety? For example, is there a way to deliver certain treatments in a way that is bringing additional layers of safety to the patient, because right now that is one of the paramount concern of our patients. The truth is basically when you check off the safety, then nobody really pays too much attention to it. But in aesthetic space, we still have significant safety concerns. So the answer is, as you can see, is multifaceted. The one segment that you would focus on is indication. From the indication perspective, what is the unmet need? But the other one is, okay, even if that indication was tackled, how can we provide better and more to the patient through this particular device or the drug that you're developing?
Wonderful. Thanks so much, Jasmina. Another question for Nalanie. So Nalanie, the question is, what elements does a site need in order to prioritize a certain project? What is interesting to them? What do they really need to get excited about a trial that they're going to run?
Definitely. So paramount to that is a well-written protocol. So that will be the basis of it all. But an organized study team, a CRO that understands the indication that's being studied and is accessible and available for the sites to be able to ask questions to. Those things are all important and that will drive the site to be able to prioritize it.
Wonderful. And just a time check, it looks like we may have room for one more question. And for any questions that we don't get to today, we will indeed respond to you in writing. Make sure we answer everyone's questions if we're not able to do so live in the webinar. So don't be discouraged if your question was not addressed today. It will be addressed subsequently. So we'll take the last one for Nicolas. Nicolas, do you have any examples of 3D measurements used for primary endpoints?
Yes, this was used, for example, for volumizing product, like, for example, mid-face or lips. These are indications for which that can be used for, either, as I said, in Europe or in Asia.
Great, thank you so much. I think that's all the time we have today. We do want to be mindful of everyone's stop at the noon hour here in the eastern side of the United States. So thank you so much, everyone, for joining the webinar. Again, we hope that you were able to learn something new, maybe a few new things today. Of course, if you have any questions or want to talk more to me or any of our panelists about running trials in medical aesthetics, please feel free to reach out. My email address is here on the screen. You can also contact us through innovaderm.com. Again, thank you so much today. Have a wonderful rest of your day, and we look forward to hopefully seeing you again in a future webinar. Take care, everyone.
Take care. Thank you.
Thank you.
Thank you, Jason. Thank you, everyone.